Okay, hello everyone. Welcome to this fourth session of the 2022 Biocontrol Workshop Series between CABI and ASEAN Fall Armyworm Action Plan. We're really delighted to see you. It's going to be another great session lined up exploring baculoviruses and looking at all the different solutions within that sort of family of biocontrol, bioprotection approaches to control fall armyworm and other plant pests and diseases. I'm Alison Watson. I'm going to be moderating the session. It really is a delight today. I've seen the presentation, a bit of a preview briefly, and it's a great one that the team have lined up for you. So I'm very excited. Before I go on to the next slide, I'm just going to thank our supporters of the ASEAN Fall Armyworm Action Plan, Department of Foreign Affairs and Trade Australia, Australian Aid, also CSIRO, the ASEAN Secretariat, and the Ministry of Agriculture and Rural Development Vietnam for all your support for the action plan and its work. And a really big thank you to CABI for joining us again to actually run this series with us, because it's a very important series. We have a lot of people joining us, and we really appreciate CABI's partnership on this work. I'm just going to go to the next slide. And I'm just going to remind everyone, and actually I'm sure no one needs a reminder, but look, if you have questions, please put them in the Q&A box today. I think there'll be a lot of questions on this, so it's really, it helps us a lot to manage the session and to make sure we do get your questions asked. If you do put them in that Q&A box, it's a lot easier. But please also use the chat box a lot, because it really helps to sort of drive a discussion. It grows our community on biocontrol, and you'll actually have the chance to probably talk to others that have got a real interest in the same work as you as well. So it's a great networking opportunity. Just a reminder for everyone, the session will be recorded and a copy will be posted 1 week after the session. You can see all the previous videos and PDF copies of presentations on the videos page on the ASEAN Fall Armyworm Action Plan Knowledge and Innovation website. Next slide. Okay, so just a reminder, today we're at session 4. We've got a whole lot of other sessions coming up, so don't forget to join us for those sessions. And there's that web link there that I just talked about. If you want to register, it's at the events page, and you can go to the videos to watch the sessions. Next page, please. Okay, I'm going to launch a quick poll. It's a good chance just to warm everyone up. And let me just get into the big screen. And also just find out a little bit about your knowledge on the subject. I've launched it now. There's 2 questions. They're sort of a little bit the same, but they've got a slightly different focus. One is, have you worked with or researched nucleopolyhedroviruses, or do you know a lot already about MPVs? And the second question is, have you used baculoviruses to control pests on a farm? So there's a slightly different focus there, and we're quite keen to see what the mix is in the room of experience with this particular set of solutions. Hi, Hong from SCV Vietnam. Nice to see you. And we've already got 65% of people who have participated in the poll. I'm going to see if I can get up to 80%. Which is exceptional. We're almost there. So if you haven't done the poll, please do so, because I'm going to close it very soon. I'm going to see if I can reach that magical 80%. Oh, I have. So virtually everyone in the room has participated. So I'm going to end the poll, and I'm going to share the results. And they're pretty interesting. So have you worked with or researched MPVs, or do you know a lot about this subject already? We've got 69% no and 31%, a good third almost, yes. So that's going to be really interesting. I think we're going to have some really specific questions there to the team, and probably some other broader questions as well, but that's actually really interesting, and it's going to be quite a good discussion, I think. For question 2, who has used the baculoviruses to control pests on a farm? You've got 20% in the room that have done that. So that's actually exceptional. And 80% then no. So I'm sure they're looking forward to finding out what happened in the field trials, for example, in Malaysia and Indonesia and in other countries around the world that the team will be sharing with us. So thanks for participating there. I'm going to close that down. I'm going to ask if Philip could move to the next slide. We've got a great lineup here from AgBTech. We've got Shashi Gurumayam, Head of the Business Development. We've got Philip Armitage from Australia, who's the General Manager there. And we've got Dr. Fahim Muhammad, who's the Integrated Crop Management Advisor at KEBI Malaysia, who will be talking later on about some of those field trials. It's a great lineup. We're really excited to have you here to talk about the subject and also to talk about your experiences with using Forlagen and also your experiences in the field and how it's working and what your plans are for the future. So thank you so much for joining us. We're really excited. And I'm going to introduce Philip because I think Philip's going to be our first speaker today. Just remember, if you have questions for Philip, and I'm sure you will, and make sure you question him lots because he's here in the hot seat. He's ready for anything you throw at him, and Shashi's there to help out as well. So take the opportunity, put those questions in the Q&A box, and we'll make sure we get them answered through the session. Philip, welcome.
Thank you very much, Alison. I think I'd just like to start off by acknowledging all the traditional owners of the lands in which we're meeting around the world. It always astounds me as to the many different countries that we're all from. With regards to baculoviruses in Australia, we're very lucky because we have an extremely high uptake of baculoviruses. So we have quite a wealth of experience with Helicoverpa baculovirus. Over 80% of the sorghum in Australia is treated annually with a baculovirus. And based on our numbers, I think nearly every hectare of maize and sweet corn has been treated with the Spodoptera frugiperta baculovirus this year. So we've got a lot of information that we can share here from Australia. Sachi's just asked if I might lead off a little bit with regards to the background about AgBiTech. After the 1990s, with the massive onslaught of resistance in cotton and prior to the release of BT cotton, in Australia we had huge problems with resistance in Helicoverpa. There was a large push to try and commercialise Helicoverpa MPV in cotton, which was met with a very poor levels of success. But going into the early 2000s, with a farmer and a couple of technical people, there was work done to actually set up and commercialise an Australian strain of the baculovirus and mix it with the North American strain. And that was very much the beginnings in a farmer's shed, literally with Mr. Bidstrup's wife's food mixer making the diet, that AgBiTech started off. And now globally, we're on most of the continents around the world with a very large production facility in the US. But it all started in Australia, where we had our large production facility prior to having the one from the US. This is a very, very busy slide, but the take home from this is with the evolution of baculoviruses, it was in the early 2000s, 20 years ago in Australia, where AgBiTech kicked off. There was work done prior to that. But in around 2014-15 was a change with AgBiTech where the majority ownership of the company was actually sold to a venture capital company to try and generate enough capital to get scale with the idea of commercializing in North America and particularly South America, mainly Brazil. So as you can see, there's a raft of baculoviruses there. We've got a very deep pipeline of new baculoviruses. The manufacture of the baculoviruses is actually very simple in some ways. It's akin to just infecting lots of caterpillars and then harvesting what comes off from that, and that's then processed for in the field. The real cleverness comes with the mass production systems of the caterpillars. The more caterpillars you can produce and the cheaper you can produce those caterpillars, the more cheap you can actually produce, the more cost effectively you can actually produce it. And as we've discovered, all these species of caterpillars, whilst they seem very, very similar, have got extremely different issues when you're actually rearing them en masse. The production facility in the US and the facility we had here in Australia, we're producing a bit over 1 million caterpillars of any one species per day. That has its own significant challenges. The product range then is covering off primarily across fruits, vegetables, soybean, dry beans, fruit and vegetables, peanuts, cotton, corn, pasture, dry beans. We're not going to cover this off today. As a company, whilst we're focused primarily on baculoviruses, we're moving very quickly into the use of semiochemicals. The semiochemicals are used to change the behavior of insect pests, and we've got an enormous development pipeline at the moment looking at how we can actually work with Spodoptera frugiperta, given the fact that we're working very extensively in Australia with Helicoverpa primarily in cotton with their semiochemicals. So I just wanted to touch on this pest. It was in 2020. This pest arrived in the north of Australia. It came across from Africa via India, China, Southeast Asia, and then came in the very tip within Australia. And the landscape here was extremely benign when it came to the introduction of this pest. I just wanted to talk briefly about some fundamentals that we've discovered about this pest, and it goes back to the fundamentals, and this will seem quite obvious, but there are two primary survival mechanisms that a lot of these noctuid moths or these Lepidoptera moths do. The first one is very high fecundity, and the second one there is their ability to be able to meet toxicological challenges because they've got an extremely diverse genome. If you were to take that Spodoptera frugiperta female that was on the previous screen, she will lay as a moth approximately 1,000 eggs over probably the first 10 days of her 2-week life, depending on the climate. Now, if we were just to take a number of those eggs, let's say 16% of them manage to survive, just for the argument's sake of doing the math, this will give you, given half of them are males, you can get an increase of 80 females from one female in a 25-day period. So we're looking at 4 generations within 100 days, which seems to be roughly the span of a maize crop. So if you were to take that kind of survival rate, if you take 80 times 80 times 80 across the 4 generations, that one caterpillar can potentially deliver within a 100-day period over 40 million offspring. This is one of the major mechanisms that they've got. Even if you halve that survival and you take it back to, say, an 8% survival, which doesn't sound like much, half those are going to be males, let's not worry. 4 generations, 100 days, that will still give you 2.5 million offspring from one caterpillar in 100 days. And even if you take that down to just purely a 4% survival, you end up with 20 females per generation. That still comes in at 160,000. So even with a 4% survival, these species have got this incredible ability to be able to produce lots and lots of offspring. That's the first survival mechanism. The second survival mechanism relates to an ancient war that's occurred between caterpillars and plants. Now, this ancient war relates to the fact that the plants were on the land first, and then insects that came along ate them second. They selected for the plants that had a mutation that developed a toxin. The numbers of insects feeding on them dropped away, selected for those insects that were intolerant for that toxin, and away it went. And I'm not telling you anything you don't know, but the fundamentals of resistance are very much a very ancient system that battle between caterpillars and you've got between the plants. Every time a plant generates a new toxin or some novel means of stopping caterpillars from eating it, the caterpillars throw lots of numbers at it with a broad genetic base. And so this war of attrition goes on, this war that's continued for millions of years, an ever increasing technological battle between more and more complex toxins that the plants to defend themselves with, and more clever ways that the insects manage to overcome. And I apologize about the simpleness here, but that was the only way I could come to try and explain that resistance is something that has been going on for millions and millions of years, and we are very much latecomers to it. So some of the toxins that the plants have produced, you will know quite well: tannins, limonene, rotenone, citronella, gossypol, and pyrethrin. These are all insecticides that largely are failed insecticides. These are insecticides that are ancient weapons on the battlefield. These are the bows and arrows of a modern battlefield. Also are these 3 molecules: nicotine, caffeine, and cannabidiol. These are also failed insecticides, but by some happy coincidence, in the mammalian brain, they bind to various receptors and we enjoy them. We've selected very heavily for the plants that produce nicotine, caffeine, and cannabidiol, so we can actually enjoy those compounds. But one of the primary pests that gets into medicinal cannabis, which is producing very high levels of cannabidiol, are Helicoverpa. The levels of resistance to cannabidiol are immense. This is because the insects have developed resistance to it. So when these insects are challenged with new novel molecules, we throw at them. For example, there's some insecticides you might be well familiar with. The same weaponry which has been employed for millions of years is simply applied to these novel compounds that we apply. And I hate to be a deliverer of bad news, but all insecticides are doomed to failure because insects will overcome them eventually, given their experience and their capabilities. Even when we've taken natural insecticides that these plants have developed, the pyrethrins, we developed alpha-cypermethrin and a whole range of synthetic pyrethroids, spinosyn, which is a synthetic version of spinosad, and even when we've taken a toxin from a soil-borne bacteria and actually then thrown at these insects, they develop resistance to it. So this battle that we've got has been going on for millions of years. We're latecomers to it. We bring new novel toxins, and these insects think, well, there's nothing new about this whatsoever, and they overcome them. However, and this is the fun bit, in that battlefield, there are some insurgents, and these insurgents have been operating in that same battlefield for millions of years themselves. No caterpillar has ever developed resistance to an assassin bug. No caterpillar egg has ever survived an onslaught from a lady beetle or a trichogramma wasp. Also, we also have the pathogens, and this is our area and my area of passion, is the presence of the pathogens in this. That while the beneficial insects might have a reproductive cycle very similar to the moths of 20 to 30 days, the bacteria, fungi, and viruses have a life cycle of several times a day. So the ability for these insects to develop resistance to these natural occurring pathogens is very, very limited. And this is why we hold up so much hope that by taking a biological approach that does no harm to the insects, the beneficial insects, and uses the biological approach, tips the scales in the favor of us. I'm not saying we need to replace conventional chemistry, but keeping in mind conventional chemistry is always doomed to failure. And that's my sales pitch, as it were. So how do MPVs work? So the virus itself is ingested into the midgut of the caterpillar, where it then permeates through the insect. It's got to break through the epithelial cells on the inside of the gut. It spreads through the insect becomes quite ill and eventually dies, and then the virus liquefies the caterpillar. And that liquefied part completes this life cycle of the virus, where the virus is then spread through the environment. Because the virus has absolutely no impact on any other beneficial insects, like flies, like other caterpillars, it's spread through the environment by rain and mechanically. So by mass producing caterpillars, deliberately infecting them, harvesting the virus that comes from them, and then applying that to the crop, what we're doing is we're just simply preceding the environment with a large amount of virus that would normally break out incredibly rarely. Now, we've also discovered that these viruses are very, very species specific, and they have varying levels of efficacy in the field. So here are some examples of dead gooey caterpillars, and these are some of my favorite photos. I know some people would call me a sick man from that, but these are images of caterpillars that have died. The virus has liquefied them and then is in the process of being spread through the environment. Here is a Helicoverpa larvae eating another Helicoverpa larvae that has died. We find, particularly with Helicoverpa, and I know we're talking about fall armyworm today, the Helicoverpa virus causes the insects to be sick. They tend to climb to the top of the plant, they die, and that's where they actually then get spread around the environment from. And later on in the presentation, I'll be talking about the difference between the fall armyworm virus and the Helicoverpa virus, because they are quite different with regards to efficacy. Helicoverpa virus is the gold standard. Now, I've got two slides here, two images to show you. The image on the right are Helicoverpa eggs, which are laid on some faba beans in Australia. And you'll note, and if you've had anything to do with Helicoverpa, they lay their eggs at random in relatively safe places all through the canopy, and they're scattered. What happens is the larvae hatches off, it then eats its egg, and then it grazes around the area where the egg is laid. And this is a mechanism that means that the transgenic crops that are containing Bt is very, very effective, because that neonate or hatchling caterpillar eats as a tiny little caterpillar, ingests straight away its first meal is of a toxin. But in this environment here, they hatch off, they'll graze around, they'll then go and find themselves somewhere to live and somewhere to hide and somewhere they can feed. Now, on the left is an egg mass from a fall armyworm. These fall armyworm, when they hatch off, they're cannibalistic. They discover they've got lots and lots of siblings, and they run to get away from each other. And they'll run to the edge of the leaf. They'll then spin a thin little web and hang down from the leaf blade and hope that wind will go along and blow them off, and they'll blow away, land somewhere, and then scurry away and hide somewhere safe. Now, we know from producing fall armyworm in the factory that they are very, very light sensitive. So the reason why I'm showing you this slide is that while the Helicoverpa larvae, which is grazing and then wandering away and hiding, the fall armyworm larvae hatches off and then makes a beeline to get itself hidden. The reason why I'm saying this is that when you're trying to target that insect with an insecticide, there is very little chance for that caterpillar to actually get exposed to the insecticide, both on the leaf blade and also by it ingesting something. And this is a fundamental difference where the behavior of the insect is working against the insecticide. You can make things work in the laboratory with a bioassay, but you don't necessarily get the field results. And this is a very important point that we'll come back to later on when we're dealing the difference between the Helicoverpa virus and the fall armyworm virus. So what do these occlusion bodies that hold the virus look like? In 1 milliliter of Forlagen, there are 7.5 billion occlusion bodies. Now, these occlusion bodies are akin to a small lump of plasticine that contains the actual virus itself. Now, the occlusion body itself has to be ingested into the midgut of the caterpillar. And unlike ourselves that we have an acid gut, the fall armyworm and Helicoverpa larvae have an alkaline gut. And in those alkaline conditions, that occlusion body dissolves and releases the virus into that midgut with a pH of about a pH of 10 or 10.5. That virus comes into contact with the gut wall, and the gut wall is protected by a mucal membrane that stops the soup, which is the interior of the caterpillar, coming into contact with the caterpillar itself. The virus has got to get through that membrane and then into the epithelial cells and then spread through the caterpillar. So there's a lot of biology that's got to happen here. And the virus itself often is not working in isolation within the gut of the insect. There's usually other things that are going on which either predispose it to being more susceptible or potentially make it less susceptible. And this is part of the problem with biologicals. And as I say to the Australian farmers here, using biological products are not for the agronomic lightweight people, because they're not like a conventional insecticide you can put out there. They are tricky to use, but if you can get them to work, they are fully selective and they are completely non-toxic. So we're talking about the production of the virus rolling in the crop, and they call this an epizootic effect. Now, with the Helicoverpa virus, and the top image there is of a Helicoverpa caterpillar in a chickpea that's died, the Helicoverpa virus will infect instars right up to 5th instar. The recommendation is never target larvae which are bigger than 3rd instar, and that's primarily because the damage is already done in the crop. However, the fall armyworm virus, the Spodoptera fructiperta virus, seems to only infect 1st and 2nd instars. So if you think about caterpillars as a biofactory, if you're looking at the fall armyworm virus, the Forlagen, it's only working on 1st and 2nd instar. The caterpillars are very tiny, and the amount of biomass, or the biofactory, is quite tiny, as compared to the Helicoverpa virus, which the big caterpillars will produce 100-fold or 200-fold more virus per dead caterpillar to get that rolling infection in the crop. So we very rarely see, or we only occasionally see, these epizootic effects with the fall armyworm virus, and we think this is partially because the amount that's actually produced by the dead caterpillars is quite limited as compared to the epizootic effect that you see in the Helicoverpa. So just running through the summary here, the MPVs have evolved in co-hosting with their caterpillars over the millions of years. Once the MPVs are ingested, they multiply rapidly through the insect. They change the insect's behavior somewhat. And when they die, they release large quantities of virus into the environment. So the benefits of this are that there's no impact on beneficial species. And I will come back to this later on, that we do no harm. At the very least, we do no harm, as compared to some conventional insecticides, which are marketed as being selective, but some of them are very, very selective to all but one species. So there is no impact with that. There is no known resistance to the Helicoverpa or fall armyworm virus, particularly in Australia. I think there are the occasional report of resistance globally with apple codling moth virus, but the chances of insecticide resistance, of developing resistance, are incredibly minimal because of the very nature of how this pathogen has evolved in the environment over the millions of years with its host. And the other thing too is because these NPVs are incredibly benign, they're very easy to mix with other things as long as they are not very high in pH, for the reasons I mentioned before about the gut making the occlusion bodies dissolve. It makes them very easy to be mixed with things, including irrigation water. And I'll come back to that a bit later on. But finally, one of the key things is because they are very, very species specific, they have no impact with humans. So the re-entry period and the harvest interval periods are virtually non-existent because these things are incredibly susceptible. So I just thought at that point, Alison, I might just see if there are any questions, because I've been
Yes.
rolling on here for a little while.
Philip, just before I ask, quite a few questions are coming in, but before I ask those, great description and explanation, I have to say. It was really, really clear. It's one of the clearer presentations I've seen, although I have to say the standard's extremely high across the whole program. But I really liked how you showed those survival mechanisms, I guess, that ancient war almost between the sort of approach, and also the multiplication capability of fall armyworm. I thought that was really quite scary, actually, looking at just how fall armyworm rapidly multiplies. And it was a really good illustration there of what we're dealing with. Even if you have lower percentage of survival, is still extremely significant. So thanks for showing that. Quite a few questions are coming through, so I'll start with the first one here from Pierre. Have you ever found another virus within the mass rearing of larvae? And if so, how do you avoid it?
This is the biggest problem, and this is where the true cleverness, I suppose, of the people, the technicians that work in ag biotech, is that when you start to produce literally millions of caterpillars a day, I mean, there could be anywhere of 10 to 15 million caterpillars when you put them together, all sorts of things break out, including other diseases, fungal issues, bacterial issues. There's a whole range of things. So yes, we do. And hygiene is the absolute critical part, particularly between the side where you're infecting the caterpillars as compared to the side where you're producing the caterpillars. You have the factory runs completely two separate systems where nobody goes from one side to the other, because it only takes a couple of occlusion bodies to get into your colony, and it just wipes it out. The same as smokers as well. There are lots of diseases which can be brought through, which come through from tobacco, that can be brought in as well. So hygiene is absolutely critical as part of it. Yes, we deal with it on a day-to-day basis, and I could tell everybody, but sadly then everybody would have to die because it's our secrets. Sashi, would you have some comment on that? Yep. Yeah.
Thanks, Philip. I was just going to say, I mean, that's our fact that we can produce millions of these caterpillars, infect them, and multiple species at a time.
Yep, excellent. Thanks, Chachi. And another question here, and this sort of comes up quite a bit, actually. People sort of ask this in different ways, but is there a possibility that the viruses, after infecting fall armyworm and released in the environment, mutate and become harmful to other insects?
We haven't seen anything on that front at all, because these viruses, and particularly the Spodoptera-based viruses, are incredibly species specific. To actually get them to jump species is virtually impossible. I haven't heard of anything in that space whatsoever. And these are, I mean, there's been subsistence farmers have been using these dead gooey caterpillars since the 1980s for producing their own products. So they're incredibly benign, much the same as dog and cat flu won't jump species to humans for the same sort of reasons.
Yeah, okay. Here's a good question for Southeast Asia, because it's a tropical region. Can you please guide about the formulation of these viruses for high temperature regions, or can you give a bit of is there some sort of difference in the according to the climate that you're applying these NPVs?
So maybe you can answer that during the data discussion, Philip.
Okay, excellent. Yep. Let's keep that question lined up then. Now there's quite a few other questions here. Somebody from I think Myanmar it was talking about BT biopesticides, but I guess and I think you're going to ask this, Shashi, you might answer this later in the presentation, but around, is there the ability to trial or do research on this with AgBTech, for example, in other countries that you're not currently working? So are you going to sort of discuss, maybe Philip, it might be you, are you going to discuss your sort of plans for the future?
I think that's very much, Saki's there. He's our development person.
Excellent.
So yeah, Saki, you might be best to answer that.
Yep.
Yeah, and I'll touch on that during.
Perfect. Shachi, I'm just going to ask you to, if you may have to turn your camera off. I'm not sure if it's just me, but I'm not always getting a good feed from you. So your voice is sometimes a little bit cut up.
Turn off my camera?
Yeah, turn your camera off. We'll just see if that makes a difference. I've got another question here. You talked about semiochemicals? You're looking at that as well and working on that, Philip. Can you use semiochemicals and these MPVs together, or other bioprotection solutions? You mentioned that a little bit in your discussion, but is it sort of part of an IPM approach? You can use them with different solutions at the same time?
Absolutely. So we're currently doing that now with the magnet, which is the attractant itself. So we're actually putting magnet out in crop in a strip every 72 meters as a thick bait that's actually has a toxicant added to it. So in some ways you've got the MPV, which is not touching the beneficials, and you're only treating less than 1% of the field with the baiting. That's actually picking up the adults and dropping the number of eggs down. So an integrated solution, that's pretty much the holy grail of where we're going to with this. We're using magnet quite extensively in cotton, particularly in southern Australia. We've got over 70,000 hectares with 3 applications this year, specifically looking at managing Helicoverpa in amongst it all. They're not using NPV in that space because it is the triple gene transgenic material, Bollgard 3, but still in all, attacking the adult phase of these pests is very much, I suppose, an area that we're focused on as a company, because this is an area we've never had much luck with, and we've got some tools in that space.
Great. Thanks, Philip. Now, we've got lots of other questions there, but I think they're going to be answered or they're going to be quite relevant for the next presentation. So I'm just going to leave it there. But if you've asked questions, don't worry. We're going to come back to them. I've already set a place for some of the questions that we've just talked about that will be answered. But I see ones about cost and also when to spray it or when to use it, practical application, sampling and threshold. So we'll get back to those questions. Meanwhile, if you do have any more questions, keep them coming because the team will be working on those as well later. If we don't get a chance to talk about them verbally, we will be answering them in writing as well. So Shachi, I think you're going next, if I'm right. Are you taking over from Philip?
Yes. Alison, I should also point out too, Ian Summerlad is our senior agronomist, is online here with as well. And he is more than versed in all this sort of stuff. So he can probably also answer some of those online questions as well as we're going. So Ian, sorry to drop you in it there, but we have a resource for sure. Yep.
Great. Well, I'm just going to upgrade Ian then as well so he can answer some of the questions in writing. But Shachi, go for it. So you start.
Thank you. Right. So one of the questions was related to cost of production. And the good news there is, AgBiotech is AgBiotech today for the fact that we can manufacture baculoviruses at scale with consistent quality that we check throughout the production process. And when you think about it, we have a production process of a biological product that is fairly automated and highly customized. And this enables us to produce, as we said, between 1.2 to 1.5 million caterpillars a day during peak season, and not necessarily of a single species. So the fact that we can keep them apart, we can go through the production cycle and run this without having cross-contamination is in itself an achievement. That's one of the reasons why we're able to roll out a biological product such as a baculovirus on a global scale. That picture there is from our factory in Texas, fairly automated. You can see my colleague Adrian there who's running the process. And what you see, the second picture there at the bottom corner is the automated line where we carry out the infection, for example. So the way it works is we have our own moth rooms where we would also have our own egg rooms. We harvest them, we grow the caterpillars, we infect them as they grow, we feed them, and then they go through this entire circle until they die when we harvest the viruses to then formulate into products like Faulogen and Lepigen and Heligen and others. Philip, if you could move to the next slide, please. Now, because of that fairly automated process, what we've been able to do is reduce COGS significantly. COGS, or the cost of biological products, is one of the major hurdles to adoption at scale by farmers around the world. And we've been able to address a big part of that through improvements in our COGS, cost of goods sold, between 2018, 2020, partly because the significant investment that came in from Paint Swartz Partners in 2015, which then went into building a brand new factory in Texas and then improving the process there. We've made improvements around, pupae harvesting, egg collection, again, automating the processes. And then how do you ensure that every egg that you harvest yields something and yields a caterpillar? And then also how about producing the diet that results in the significant growth of the caterpillar while the virus is also multiplying inside the caterpillar, such that at the time you harvest the dead caterpillars, it's also full of viruses. We've also done a lot of studies to look at the cost of production elsewhere around the world, in China, in European countries. And we believe that we have a significant cost advantage versus our competition in Europe and in China, from the very simple explanation that I made, but it's our trade secret. We can't tell you much more, but the comparisons that we've run internally show that we do have a significant cost advantage. Again, you have to remember that baculoviruses are naturally present. They're out there. If anybody wants to multiply them, they can go, and harvest and then multiply on their own. The trick is in being able to multiply at such a scale like ours, and at a cost of production that is extremely competitive. Philip, if you could move to the next slide, please. We'll talk about Faulogen, which is the product that we have for fall armyworm. As Philip has already described, it's an NPV product. You've already heard about the mode of action, which is also a recognized Group 31 mode of action. One or two of the questions that were asked before related to resistance. I mean, there are no known cases of resistance. Remember, all that we're doing is essentially multiplying what happens in the natural world out there. We're just bringing it into a factory environment and multiplying, accelerating the process, multiplying and producing just a lot more. But we are not doing anything that isn't happening already out there in nature. Therefore, we have not come across any cases of resistance to NPV-based products in the 20 years that we've been operating, and also with publications that are out there. We haven't seen anyone reporting resistance. Our products are OMRI certified, so for anyone who is growing organic crops, they can use the products without worrying about the impact on the certification of their crops as organic. We have also seen that Faulogen can be used as a standalone product in parts of the world, and we've also seen that as an integrated pest management solution, it is a great partner. And we'll show some results related to those in a bit. Thank you, Philip. Okay, so, oops. I mean, this is a map from Kabi highlighting all parts of the world where fall armyworm has spread over the last, I would say, 5 to 10 years. We know that it was always there for a long period of time in the Americas, but then since 2016 or so, it's been spreading rapidly across Africa, maybe Southern Europe, and then towards South Asia. That's when, like 2018 to 2020, when it came in. And then it's spread across to Australasia, all the way to the Far East. And this, invasive species is now a major threat to agricultural productivity all around the world. And we want to be able to play a part in providing a biological solution, sustainable solution to farmers around the world. And one of the things that we want to do today is share some data that we've got from parts of the world to show you that baculoviruses can work in different agroecological environments around the world. Next slide, please. So we know it was there in the Americas. We've been working in the Americas with fall armyworm and baculoviruses. But from 2018 onwards, we started establishing partnerships with the likes of UPL, Flour Mills of Nigeria, Kabi, of course, as our key research partner. At the time, it was in Kenya, USAID with the support of the FAO and so on in Africa, where we started rolling out Phalogen to various countries, including Nigeria, South Sudan, Kenya, Ivory Coast, and so on. At first, by just running local evaluations, then formal registration trials, and then eventually releasing them in the country for commercial use. We also established partnerships with Ispahani in Bangladesh with the support of CIMMYT and USAID. You'll see, the running trend here is that it's always a public-private partnership. We are a relatively small company, and therefore we do seek help from international organizations and local partners to take our products to farmers in those countries. We also have a partnership in Sri Lanka with a company called Bowers, which is a Swiss legacy company, one of the largest inputs distributors in Sri Lanka. And through that, we've been able to release Phalogen in multiple countries, as I mentioned before. We've also had the support of our partners to feature Phalogen at key events so that there's more awareness, on television, on radios, and so on. And then Phalogen spread to Australia. This is where Philip will talk a bit more about some of the work that he's done in a few slides later. So I wouldn't dwell too much on that. But in 2021, we established a partnership with Kabi because we'd worked with Kabi in Kenya to evaluate Phalogen in that agroecological environment. We wanted to know if Phalogen would also work in the Southeast Asian region. And who better than our existing partner, Kabi, who also had a base in Malaysia, to work with and see if this would hold true in the local Southeast Asian environment. We've also set up partnerships with companies like DuAgro in Indonesia, Leeds Agri in the Philippines, and HSC in Vietnam. So if you have, interest in those countries, we're certainly in the process of registration. As to other countries that we haven't targeted, it's not because we're not interested, it's more to do with our ability to resource and run multiple registrations in multiple countries all at the same time. Philippe will also talk a little bit more about how farmers are adopting Phalogen in Australia. If we can move to the next slide, please, Philippe. Right, so what about performance? What we've done is in all of these countries, we start out with an evaluation, a local evaluation to see if it works or not, and then we go through the formal registration. And in Bangladesh, what we have here is the yield in tons per hectare, comparing the Phalogen treated farm with an untreated and a locally available insecticide, Coragen in this case. And you see that, it's comparable to existing solutions, if not better. And if you follow the protocol, which is around, targeting and spraying Phalogen at a time when the pest is most active, so that would be at sundown, maybe until early morning, not at midday, because as Philip's mentioned early on, they are light sensitive. They tend to hide during the day in the whole of the maize. And then, with our most of our smallholder farmers, they tend to spray with a knapsack sprayer. They tend to direct the nozzle directly into the whorl of the maize plant. So that also helps to ensure that the baculovirus gets into a point where it would be easily ingested, and then it can start doing its work, as Philip described earlier about the mode of action. In Kenya, where we've run this trial, we see that the infestation level is significantly lower versus the untreated. In Sri Lanka, we got this very nice picture of a farmer getting 500 kilograms per hectare on the untreated control versus 3.1 tons per hectare. You can see the difference there, and also look at the uniformity of the cob. The farmer is just happy, very happy farmer on the back of these results. The picture there is the gooey caterpillar that you've seen before too. But being cannibalistic, fall armyworm would attack a healthy fall armyworm would attack a weaker fall armyworm, and in doing so, if we were to attack an infected caterpillar, it would also get infected. And then as they come to munch around, this dead caterpillar here, if they were to munch around and eat the leaves with the liquid, they get infected too. And then that multiplication continues. Of course, this is more profound. This epizootic effect is more profound in Helicoverpa than in Spodoptera, from what we've seen out there in the field. But the good news is the product continues to work in Bangladesh, in Kenya, in Sri Lanka. But what about in Southeast Asia? And that's what's on the next slide. Right, this is just, sorry, before we jump to Southeast Asia, this is just looking at one of the other things that I mentioned on the integrated pest management program. So what we've also seen in Brazil is that when you mix baculoviruses with an existing standard chemical, it enhances the performance. And we believe this is because either the resistance is being weakened through the baculovirus, or the ones that have not been affected because of resistance are being targeted by the baculovirus for which there is no resistance. And so what you see there is that versus the untreated, or Metamil in this case, the standard chemical, you get a significantly better performance when you combine Cartogen, which is the brand name for Foligen in Brazil, with the chemical. And this is true in corn and cotton both. So what you see there is that, there's an improvement of 67% in corn, and we get close to 26 bales per hectare extra when you combine the treatment for cotton. This is what we call the protector concept, first, I think, proposed by colleagues at UPL. Okay, thank you, Philip. The next slide, please. Okay, continue. You can take over.
Okay, thank you, Sachi. We're moving on to Australia here now. So, yes, so as Sachi says, there's we're doing lots around the world. We are very lucky here in Australia that we've actually have a partnership with one of our public sector research agencies that are based here in Queensland. They've assisted us with getting the application to bringing the virus into Australia. Australia, as you may know, has very strict quarantine laws, but also assisting with actually identifying some of the fundamental items of the insect. The chart we've got on the screen there, and I've just been answering some of the questions in the Q&A, and this relates to it. The chart there is actually showing here the days after ingesting the virus and the percentage mortality by instar. And here we're seeing sort of somewhere between 80 and 90% mortality by about day 8 or day 9 after ingestion of first and second instar. Once you get to the third instar, you start to see the efficacy drop off significantly, and then fourth, fifth, and sixth instar, it drops off considerably. Some of the work that was done with bioassay in the laboratory went out to 12 litres per hectare instead of 200 mils, and sixth instar larvae were still very tolerant to it. So that is an issue there. We need to target these very small larvae, which is quite tricky in itself. I should point out then, that the susceptibility to the fall armyworm virus is different between Australia and Brazil. We've got a large laboratory of our own in Brazil, And these results that we're getting here, our Brazilian team were quite excited about, because they don't quite see that same level of efficacy, particularly on the first and second instar larvae. So genetically, and this is the work that the CSIRO have done, I think we're looking at, and other research agencies, I think we're looking at perhaps 1 or 2 or even 3 separate incursions from Central America into Brazil, and then out of Brazil across, out of Brazil into Africa, and then from Africa through to Australia. So the population we have here in Australia is relatively quite removed genetically from the originating population in Brazil. One of the things that some of the research is showing, and we're finding this in Brazil as well as we're finding it here in Australia, is that there's some tantalizing information that when you're introducing the fall armyworm virus, Forlagin, into a production system with conventional insecticides, at the end of the season, there is less damage, although it's harder to see the performance of the virus itself. There is something that's going on there. I wouldn't go as far as saying that there's synergy or those sorts of things, but there is something potentially in there which is improving either the performance of the insecticides or improving the performance of the Forlagin on the caterpillars. We don't know, but it's very useful. And this is the exciting bit for the future. As I mentioned before, using NPVs in an agricultural system can be tricky, and this is part of the challenge, and this is why we're having so much fun. One of the things is we tend to look at insect control based around the application of an insecticide or something that kills them. The reality is, when you're dealing with a biological system, is that we are dealing with many other species of insects. So at the very least, the virus is going to do no harm to any other insect, and at the very best, it's going to complement them. So when I'm working with farmers here in Australia, I tend to be explaining to them that the virus is doing some of the work, and the beneficial insects are doing a lot of the work. When we first had fall armyworm strike here in Australia, we collected larvae from an area about 500 kilometres south of where we are here in Toowoomba. The larvae were collected and they were sent to the laboratory here in Toowoomba. And of the 38 larvae that were sent, and this is in the very early days, 36 of them were parasitized by such time as they had arrived and then incubated out. So this is showing the fact that the beneficial insects are doing quite a lot, and the virus is physically helping them. One of the other things is that, as I mentioned before, with the very high level of adoption of Helicoverpa MPV in Australia, particularly on the broadacre row crops, there's a very high level of expectations around performance. The Helicoverpa virus is the gold standard. It is bulletproof. It is reliable. It handles high heat. We know that Forlagin is nowhere near as robust from this. So one of the things we had to do here in Australia was actually manage down expectations, because people said, oh, great, I've now got a Vivus, which is the commercial name for the Helicoverpa virus here. I've now got a virus for fall armyworm, which we had to say, no, well, that's not exactly the truth, because it doesn't perform similarly. So if you remember, I was talking about before the behavior of the insects, the hatching off, hanging on threads, getting blown in the wind, getting away from the light, getting themselves entrenched. This really gives quite a challenge to present something to the caterpillar that they have to ingest. There's very limited amounts of getting an NPV into their digestive tract, as is there would be with any other stomach-based poison like a BT, for example. They're all hard. One of the things that we've discovered is that although it's very dilute, you can actually utilize MPVs through overhead irrigation water. If the irrigation event is less than a 10mm watering event, you can actually insert the virus into the irrigation water, and the whole plant gets dosed with a very, very low level of the concentration. And the numbers are somewhat, if you remember, 7.5 billion occlusion bodies per mil at 200 mils per hectare in a 10mm watering event. That is still 11 million occlusion bodies per litre of water. There's two images we have on the screen here. I was having a play around with some blue dye through a spray rig, and you can see the blue dye on the outside of the cob and on the silks there. And then on the right, I was quite astounded when I actually ran it through a simulator for a 10-minute watering event with the blue dye. As you can see in that image, the blue dye has permeated completely through the plant. And that then struck me is that all those places that the larvae like to go and hide, such as in the leaf whorl, where the leaf blade attaches to the stalk of the plant, that's where the larvae are going to hide, and that's where they first feed. If you can actually present the virus to them in those places where they were feeding, then you've got them. And this is why we think that the water run overhead irrigation work is working so very, very well. In fact, on the numbers I have at the moment, we're up to more than one full spray across every hectare of maize and sweet corn in Australia has gone out either as a mix with another insecticide or is going out regularly every 7 to 10 days through the overhead irrigation. So take that as it may. I think this also reflects Saatchi's comment with the results he's seeing in Africa with the hand applied material, where it's actually being applied down the whorl of the plant. The product is ending up down into the whorl. The two things that break down the occlusion bodies, one is UV light and two is high temperatures. So inside the plant, the temperature is at 22, 23 degrees and it's dark, and that's exactly where the caterpillars are. So this is where we're thinking that there are some benefit to be had by considering the application of it and using an overhead irrigation in somewhat like a super boom spray. The work is also a difference in Australia between Australia and Brazil with regards to the rate response. In Brazil, we tend to find a fairly flat rate response in the larvae between 50 mils to 200 mils per hectare. That work was replicated here, and it would seem that it is different. Initially, we were looking at a recommendation of 100 mils per hectare, but we've seen in Australia a dose response stepping up from 100 mils to 200 mils to the hectare. Again, showing you the difference between the respective instars, but you're finding that there is a slightly higher rate is giving you a better result. This is the work that's been done by the Department of Agriculture and Fisheries here in Queensland, per courtesy of Dr. Melina Miles. And the other thing, which is perhaps not quite so relevant to Southeast Asia, but it is relevant here, is temperature. You have to remember that fall armyworm are a tropical pest, but they do persist into the temperate areas. And it's called fall armyworm because it breeds up in the Central Americas in the spring and in the summer, and it's quite sedentary. It only disperses. But then in the autumn or the fall, something triggers it, and it might be related to day length, to go into migration in the fall. And that's why it's called the fall armyworm. And it will move as far north as the Canadian border on the North American continent and the bottom end of Argentina on South America. So this is a trigger. So this graph here is showing you the response of the time to death based on temperature. So at 35, 30 degrees, and even down to 25 degrees Celsius, that mortality after ingestion happens quite quickly. However, once the temperature starts to fall below 20 degrees C, that time to death is considerably slower. Now you have to remember, the virus itself is inert, it isn't alive, it relies on the biological processes within the insect to multiply it. So as a result, if the insect is not thriving, if it's struggling in cool conditions, it's not growing very quickly, the virus is not propagating through the insect terribly quickly at all. So these are some of the key things that we've actually learned here, and we are continuing to work with it at the moment. We're very encouraged by where we're at, but I keep coming back to the fact that it is tricky to use, but the rewards, if you can get it to work, are absolutely there, both human toxicity-wise and also performance in the field. Saatchi, I'll go ahead and back over to you.
Thank you, Philip. I think I'm going to try and turn on the video. Tell me if the network is not great, and I'll switch it back off. So I think, we've heard about the performances around the world. We wanted to know if the agroecological environment in Southeast Asia would also be conducive to the performance of Farligent against fall armyworm, similar to what we'd seen elsewhere. Of course, we talked about temperature being, high temperatures being a risk and exposure to UV light. But then again, if you work with the farmers and if you're able to educate on how baculoviruses work, the time of the day when they should spray, how they should spray, and so on, which is the how being, directing the product directly into the whorl of the maize plant where the caterpillars are hiding, and so on, then you can actually replicate the good performances that we see elsewhere. And that's what we've done here in Indonesia. Fahim, our colleague from Kabi, will talk about the results from Malaysia, but I just want to explain this particular chart from Indonesia so that you understand what we've done here. We basically went and worked with a farmer who had his own standard practice. We didn't have an untreated control because the farmer didn't want to lose any of his crop. And we compared it against 100 mils per hectare of Phylogen. We chose 100 mils because we know that 50 mil, even though it might work, does not give sufficient coverage, and it may not be enough to cover the entire crop, even though we have a very high concentration of OBs per mil. What we've also seen in other countries is that between 100 and 200 mil, Philip is seeing slightly different results in Australia, but between 100 and 200 mils, we don't see a significant step change as we do between 50 and 100 mils per hectare. So we chose 100 mils, which is the standard recommendation for Phylogen, and you see the performance there. The other thing is we recommend between 3 to 5 sprays, weekly sprays, from the moment you see a critical threshold of infestation. So that's something that we're also trying to do, you know, scouting, understanding how the pest spreads, infests your crop. You have to scout regularly and understand at what point you need to start spraying. So from the moment you start spraying, we say 3 to 5 sprays, weekly sprays. And of course, you have to keep scouting. You have to check the damage because fall armyworm is extremely mobile. Even if you're able to control all of the pests on your plot, it is quite likely that if your neighbor's plot is untreated, they'll spread from there overnight, fly in, you know, spread through the wind, crawl over, whatever, but they will spread. And this is something that farmers have to be extremely cautious about. What you do see here is that the local farmer standard, which is an emamectin-based product at about 250 mils per hectare versus Phylogen 100 mils per hectare, you get a very similar performance. You know, 15 days, the first bar that you see there is before application. The rest are after 25 days, 35 days, and 45 days after application. So sorry, this is after planting. The result in Indonesia is giving us confidence that the product does work, even in the Indonesian environment. Now, remember, we set up the partnership with Kabi because we wanted to check with a dedicated research partner in that agroecological environment. If Kabi had been present in every country in Southeast Asia, probably we would have gone to every country in Southeast Asia to run trials with them. But we felt that, you know, running it in Malaysia would also be representative enough for the other countries in the region to give us enough confidence to then go and start trials in Vietnam, in the Philippines, and so on. And we've seen, even in the Philippines, where we've now got some early stage data, that the performance is replicated. So that's the good news here. Okay. Philip, if you could move to the next slide, please. Right, Fahim, at this point, I'd like to request you to take over.
Okay, thank you. So as Mr. Sachi told, so we are having a partnership with Kabi has a partnership with Agribiotech for seeing the effect of this Phylogen product in the Southeast Asian environment. So we started these initial studies in our Kabi Malaysia office. We did some bioassay studies in the incubators for fall armyworm on the diet. And the initial results of these bioassay studies is quite a very positive one. So we can see if we tested three different doses, 100 as a standard, and 50 as half of the standard, and 200 is the double of the standards. And if you can see here, it's almost giving a similar results like Australia, starting from the mortality is increasing from day 5 or onward, but up to the day 10, so we get a very good results of about 90% mortality in the cumulative mortality for the fall armyworm populations. So the N number of larvae is 120. We tested in this diet bioassay experiment. Philip, can you go next, please? So yeah, in this diet surface bioassay method we used for testing the foligen against the fall armyworm. So where we use the artificial diet and treated the foligen product on the surface of the diet and release the second instar larvae. So as Philip studies and other studies have shown that, so they are very effective on the second instar of the larvae. So we tested these on the second instar of larvae in the incubator studies with 28 degrees centigrade and plus 14 days of light and 10 days of dark photoperiod. So we see, you can see the pictures here. So a very good results around after 9 to 10 days, there is almost the larvae are completely infected and we get a positive results in that. Okay, can you go please next? Okay, may I request to play this video? Yeah, and okay, if you see here, so the first larvae is the control where we didn't apply anything, and the second larvae is the half dose. This is the results of the 7 days after the 7 days of bioassay study. So here you can see in the third larvae is the treatment number 3 is 100 ml dose. This is the standard one, and the last one is the last one is about the dose of the double dose. So you can see here, I think it stopped, video is stopped. Yeah, so and this is a way, actually this is in the Petri dish, but this can be the case also in the fields as well. If these healthy larvae are in touch with the infected larvae or eating on the infected larvae, so they becomes a source of a biofactory for these viruses, as rightly said in previously in by the Philip as well. So yeah, that's why these baculoviruses are giving us a very good results for the management and also for further multiplication of these baculoviruses within the field. Next, please. Next. Yeah, just aside from the fall armyworm, so we are also evaluating another product from AgBiotech is about the Lapigen. Lapigen is specialized for the diamondback moth on the cabbage. So we are testing this product in the Cameron Highland in Malaysia, so on the cabbage crop. So actually this experiment is already in progress, and we have some initial observations and have the data. So maybe we will get a chance later sometime to present that one. But the initial observation shows a very good results as compared to the untreated control, and also slightly better to the farmer practices where the farmer is applying almost 2 times chemical spray in a week on this cabbage crop. Next, please. Yeah, here just a picture demonstration of the damage caused by the diamondback moth larvae on the cabbage, and on the right side we are showing how the Lapigen is spraying on the cabbage plants. And then in the middle, so this with the red color borders, the picture shows the infected larvae of the DBM. So yeah, so this is one important thing here as well regarding the temperature thing. And in case of the cabbage environment, so when we are having the spray, so this chemical, because remains in the head when we are spraying, and the nozzles keep these viruses almost, because in the cabbage all the leaves are centeredly open, so there is a pockets is there, so where this Lapigens remains there with less effect of the temperature. And we see there is a very less damage is on the head where we are applying the Lapigen. So that's also one of the observations, but when we have the results, we can further discuss on that. And in this project also, we are testing 4 different dosage of the Lapigen and see, okay, which one is the effective in this environment. Okay, over to you, Sachi.
Thank you. Thank you. I mean, this DVM work that we're doing in Malaysia is extremely exciting because, you know, it's probably one of the pests that has developed the most resistance, short life cycle, and cabbage, brassicas are grown all over the region. So we're quite hopeful that this is going to deliver some strong differentiated results that we can then use to register Lapigen across the region. But you know, going back to what we spoke earlier about some of the changes that are required in terms of farmer practice to be able to use baculoviruses well, but at the same time to be able to recognize and understand the behavior of the pest and then to use the baculovirus in such a way that it attacks it at the point where the pest is most affected. Now, one of the things that we've done is work with SAUBO, which is Scientific Animations Without Borders. If you haven't come across them before, please go to their website, saubo-animations.org, and you'll find tons of videos related to all kinds of pests, and you know, some of them are related to fall armyworm in multiple languages that you can then download and share on your mobile or send the link to others. What we've done is work with SAUBO with the support of USAID and other organizations. We're working with the University of Purdue here to develop multiple videos. One of the videos that had already been developed was on identifying and scouting for fall armyworm, so that you can assess the level of infestation and the timing of spraying an insecticide such as Foligen or a chemical insecticide that you might find in your local input dealer. The other thing that we've done is come up with a video on controlling fall armyworm using Foligen. So it describes all of the required, you know, practices around how to mix, how to spray, when to spray, and you can see that in that picture. It's almost at sundown where the farmer is spraying. Please, I mean, if you get a chance, please go to the website and have a look. You can download the video already. The Foligen video is in a couple of languages now. We are translating it to 2-3 other languages in the countries where we do expect to launch Foligen in the coming 3 to 6 months. And by doing this, we're also experimenting, trying to see if farmers could be trained at scale using, you know, training videos like this, where, you know, they can spend time on their own in the evening after they've been to the field or early in the morning and train themselves to use the product properly. So then, you know, our responsibility is ensuring that the product is at an outlet close to where the farmers are, and then they can use the video training to work through the steps of using Foligen properly, instead of having to rely on, you know, maybe an extension agent coming out a few times in the season. So it's something that we're working on, and we're hoping that we'll have some good comparisons between farmers who are trained using the video and those who haven't watched the video in terms of yield differences and so on. And we hope to share more of that later, you know, next year or so.
1 minute, Shashi.
Yeah, if we could go to the next slide, please. We're almost done. Right, so this is the second last slide. I mean, one of the things that we've also done is look at the impact that we have on the various sustainable development goals. Of course, we are working towards creating a safe and nutritious food ecosystem. We're also making it safer for farmers because we have a product that's benign pretty much to everything else except for the very specific caterpillar that we're targeting. And therefore, we're also helping manage resistance and at the same time reduce chemical use and so on. We've also done cradle to customer analysis where we've seen that our products, our biological products, reduce greenhouse gases by 30 to 50%. And when you think about the millions of liters that are now being used around the world, it's a very significant impact. We've already talked about how our products have no impact on biodiversity because it is extremely selective. It's going only after the pests, the concerned pests. We believe that, you know, we have a product that is extremely environmentally friendly, safe, and helps us to meet many of the sustainable development goals that have been identified. I think we're done with that. The last one is just a thank you slide. Thank you.
Thank you so much. Great presentation, and I love the team approach as well. We've got quite a few questions. I know some of them, most of them have been answered all in writing, but I would like to have sort of maybe just a bit of time to discuss those so everyone can kind of see the answers, and it maybe might prompt some other questions as well. So I'm going to sort of go through quite quickly, and I'm not sure, Shachi, you can decide who from the team can answer maybe each question. But I do have a question for Fahim, and that was around, it was a comment in the chat just around, are they just laboratory testing evaluations at the moment? I mean, you have quite high, obviously great results there, but sometimes it's a different case when you get out in the field. Can you comment on that?
Yes, correct, Alison. Yes, this is the first lab bioassay studies in the incubators. Yes, you're right. So we are further planning to have this in the field as well, because and at the first level we want to see the results in the lab stage, but the populations are from the field, and no, we are planning to have these trials in the field as well and getting the results from there. Yeah. True.
Great, excellent. Thanks, Fahim. Shachi, I've got a question for you. You talked about SDGs and all that sort of broader sustainability focus, which is great. Fantastic around that climate footprint work there. What about, I guess, circular economy, plastic or recycling, the containers that Forlagen comes in, for example? What does a farmer do with those once they've used it?
Right. Thank you for asking that, Alison. I mean, this is where, you know, the further down the chain it goes, you know, you depend and rely on local practices, on recommended practices, on what a farmer should do with the empty bottles and the sachets and so on. Now, of course, there are recommendations from the industry, from the chemical industry around what to do, how to dispose of safely with, you know, the bottles and the sachets. And we do follow those. Of course, the difference is that in our case, we have a product that is benign. It's not toxic. It's not hazardous. And the management of those bottles is different. However, we would still recommend farmers to wear PPE, to dispose of the bottles as per the regulations on the ground. This is an interesting question as well, because it has a much broader industry point, which is, you know, how can the whole industry, biologicals and chemicals, work together to ensure that bottles are disposed of safely?
Great, thanks. Great answer. We had a question here around the practical application and date of spraying. What about sampling and thresholds? And some other people have asked similar types of questions. Can you give? I see Philip answered it in the chat in the question and answers, but if one of you could just maybe give a little bit of more, I guess, feedback on date, practical application, sampling, thresholds. Briefly, I know that could be a whole 60 minutes.
Is that sampling for the pests or a threshold for infestation?
I think probably a little bit of both, actually.
Okay. So there, I think the best recommendation I can give is actually that video that was developed by Salvo. If you go to the site, you'll see, you know, we have the video where we talk about how to scout in a W shape and pick you know, plants randomly and see how many are infected. And normally what we say is if 20% of the plants are affected, and with fall armyworm, you have to look at the effects. You can't really see the caterpillars at instar 1 or 2. You have to look at the window panes, the holes that you see in the leaves and so on. Then you can start spraying. So that video describes the whole thing quite nicely.
Okay, great. And I really like the emphasis on good scouting because that is one of the key things for fall armyworm management. Here's a question from Wilma. What is the shelf life of Forlagen and how do you store it to make it last long?
Do you want to take that? Or
Well,
Ian?
I'll grab this one. I just want to, if I might have, just go back to that previous question, Alison. Ian's got some insight into the field thresholds because we're talking about maize and we're talking about sweet corn are slightly different with regards to this. So, Ian, have you got any comment in that space, mate?
You just have to unmute yourself, Ian.
You're on mute, Ian.
And you're still on mute for some reason.
Maybe you can take the storage question, Philip, while Ian
Oh, there we go. Ian's unmuted.
Yeah, I'm
Brilliant.
muted. So, sorry, can we repeat that again, please? Sorry, I just lost myself. I got a bit scattered. So.
The question with regards to thresholds, Ian, and commercial thresholds, per se, difference between sweet corn and maize. It's a bit of a dark science at the moment. Have you got any insight from anybody on that?
Look, it's very much in Australia. It's very much basically fly by the seat of your pants, so to speak. I use that as a pun, but it's well, not a pun, but yeah, it's very difficult with regards to how I actually manage it. The point I did want to make was, and to emulate Saatchi's and Philip's comment, the success we've had in Australia has been around coverage. So it's not so much about water volume, it's about spray coverage, getting it in there, getting it to work, all that sort of stuff. So that's what's working here. That's what we've got. So but yeah, with regards to threshold, with regards to sorghum here domestically, we don't consider a threshold. With regards to sweet corn and maize, it's very much just get on top of it, go early, go often, and get on top of the populations.
So just excellent. So then that's good. I like that get early. I think that's the key thing with fall armyworm. So that is really what we hear all the time. But Philip, just getting back to the how long does the product last as well? And just a question, and you or Ian can answer it, but how many hours does it last on the maize leaf once you've actually sprayed it? So one is around how long can you keep the product and how do you store it? And then secondly, once you actually use it, how long does it stay on the plant? And I did hear that 3 to 5 weekly sprays before. So it'd be interesting to know what are the factors that keep it on the plant or wash it away or influence how long it stays.
So the natural enemy of the occlusion bodies is UV light and heat. So at 40 degrees Celsius in UV light, it's a matter of hours, but we talk about 48 hours, even sitting in the drum at 40 degrees Celsius. At 4 degrees and below, it will last indefinitely. There are some samples which came out of the 1980s, which are still quite viable because they've been frozen. The transition occurs between 20 to 30 degrees, where it goes from months, years back to days. It's in that sort of period. So when the occlusion bodies are exposed on a leaf blade, they're broken down pretty quickly. But if you can get coverage in the places on the plant where the plant is cool and also when it's in the shade, it lasts 3, 4, 5 days.
But in Southeast Asia, I mean, we're in a tropical climate, that's probably not going to be too cool environment.
No, so again, as Ian pointed out, coverage is the critical piece. You need to get the coverage of the whole, and that's why we're talking about running it through the irrigation water. You're putting the occlusion bodies in those dark, cool nooks and crannies where the caterpillars are going as well.
Okay. Great. Awesome.
And then once it's been ingested, Alison, it starts multiplying inside the caterpillars. It does a thing of its own.
Excellent. Oh, great. Another question, Shashi, maybe you can answer this, but what kind of nozzle are you using when spraying the virus? Now, it's quite a specific question, but it's kind of an interesting one, I think.
Well, Fahim, I think you were last week in the Cameron Highlands spraying, right? So generally,
Yeah.
when we're talking about smallholder farmers in Africa and South Asia and so on, we
Yeah.
don't go down to the specifics of changing their nozzles, because the virus is just as usable or user-friendly as other crop protection products that are out there. We haven't gone to the extent of changing nozzle size to change the droplets and so on, because then it becomes too much of a hand-holding. I know for colleagues in Brazil, in the US, and maybe for Philip and Ian, there are certain things that they could do if they're spraying, aerially versus a boom sprayer or a conventional irrigation facility. But for smallholder farmers, we don't go down to the point of saying change your nozzle.
Yep. Great. Well, great. That's a good place to leave it, I think, because we are talking about smallholder farmers in general. Yep.
Yeah, maybe I can just comment on this. I agree with Sachi, because when the first time I go to the Cameron Highland, I ask them, okay, I want to do this thing. Okay, the first question, okay, how we can apply this, which machine you need, what type of requirements are there. So either we can use the same thing, same materials, and so then they feel a little bit, okay, yeah, that's fine. So the same nozzle, same machine, but yeah, there is a little bit requirement. You need to wash it, rinse it, because if the machine is already used with the chemicals and you are using the biochemicals, so properly wash it or rinse it, or maybe 1 minute through the lance and nozzles. You can throw water, fresh water. So that's only some guidance, and the rest is I think very well explained in the video.
Yeah. Excellent. Okay, great. Well, I'm going to make sure that video is, I will provide the link in the email that I sent everyone with a copy of the presentation in the PDF, a link to that, so everyone can watch it. Fantastic presentation. Thank you to all of you, and Ian for coming on at the very end there as well and busily answering questions and writing. I could see you very, very engaged there with many questions. So that's a big thanks to all the participants, because that means you were really engaged in it, and I'm hoping that it was extremely useful for you, because the information was rich, practical, really good examples, and really well appreciated. So I'd like to thank the whole team. Before we end, I'm going to hand over to Fang from CABI, because he's just going to give a very brief summary and thank you from CABI and from the ASEAN Action Plan for both of us. So Fang, I'm going to hand it to you. And from me, everyone, thank you very much and keep safe. Fang?
Thank you, Alison. I'm always amazed at how you master this webinar. First of all, I would really like to thank Alison for her hard work to get such success of a series of biocontrol workshop. That CABI is very happy to work with you, Asian Farming Secretariat, to try to promote biological control in the region. We would also like to thank AG Biotech to have entered into such a public-private partnership. That's I think it's such a good experience working with the private industry. Of course, I also say that the baculovirus products, that Labigin and Foligin, are one of the nature-based solutions. I see this a miracle from the world that, maybe from the God to give to us to control the pests, we should use them. But of course, I see that the compatibility with the chemical pesticides, there's rooms also with the parasitoids. I think some of the participants raised. So if we could put all these biopesticide products, including baculovirus and other biopesticide products, and as well as the parasitoids, as a part of our IPM tool, I believe we will be successful to control farming. Thank you, of course, to the participants for your time to attend. Yeah, over to you, Alison.
Great, thank you so much, Fang. As always, a very nice summary at the end, and it's always a privilege actually, and a joy to be working with you and your team. So thank you very much, Agritech, for again, wonderful presentation, well prepared, and we really appreciate it. Thanks to everyone. Keep safe, everyone, and we'll see you next time. Okay, bye.
Thank you, everyone.
Bye-bye. Thanks, everyone.
Thank you.